Oh, Alex, we were supposed to start here, right, with serine synthesis, but this is a complicated mechanism, okay? So I'm going to forego his doing the mechanism of this, but I do want you to see something, and you should be able to classify what kind of reaction that is happening here, okay? Glutamate has an amine. Alpha-ketoglutarate is the carbons once the amines have been taken off. This phosphohydroxypyruvate has an alpha-keto acid, and it converts into a amino acid. And so what has really happened is the nitrogen from the glutamate has been transferred to here. So what did we transfer? An amine, and so the aminotransferase, this is an aminotransferase reaction. That's what I would want you to see from this particular type of reaction, is that the amine has jumped from one place to the other, and the jump comes from, or the jump produces an alpha-keto acid, and it reacts with an alpha-keto acid. But then that leaves Alex on the hook for this one, okay? And so all right, so we have the 3-phosphoserine, which is almost the amino acid that we want, and we have water. And so the base is going to deprotonate the water, and we can attack at the phosphate. But we attacked, remember, a phosphate that has at least one double bond. What do you have to do when we attack the double bond? Kick up, and then kick down. All right, that is one way that the mechanism could have been done. Alex, can you use another color and show me where the other mechanism you could have written and it had been 100% right? Still deprotonate. There. Yes. That results in the same product, a hydroxide attached to that carbon. Thank you, Alex. You get to select somebody. All right, the blue carbon, or the blue arrows represent what kind of mechanism? Is it E or is it S? S. I substituted something. I substituted a hydroxide for a phosphate. The phosphate was there, and I substituted a hydroxide into there. Is it written this way, an SN1 or an SN2? The blue is an SN2. Okay? Now, the green is what actually happens. Now, you wouldn't know that unless you labeled the oxygen in the water and found out where it is found in the product. If it is in the phosphate, then it attacked the phosphate. If it is in the serines, in the serine. I would have accepted either one of these answers because they're both technically organically correct. The enzyme could have done both of them. All right, and so there was two answers that I would have accepted. That doesn't violate anything that you have learned as being, that doesn't look good in a mechanism. All right, we have now synthesized serine. All right, the aromatic synthesis, aromatic amino acids are not done within the body, okay, with the exception of when phenylalanine is degraded, we can make tyrosine, but we're going to make the de novo synthesis of tyrosine here, and we're going to look at how it happens in E. coli. All right, anybody ready to jump on this one? Not really. Okay, I'll do this one. This is a little bit more complex than we've seen up to this point in time. Now, here we go. What pathway are we here if I see this? Pentose phosphate pathway. What pathway am I here? Glycolysis, or you could have answered gluconeogenesis. Okay. All right. Looking at this, we've got a lot of carbons. All right. At this point in time, we need to do some inventory. So I'm going to do this. This red phosphate is that red phosphate. followed by that oxygen, this methylene, that methylene, this hydroxide, that hydroxide, this hydroxide, that hydroxide. This carbon would be that carbon. Okay. If I go over here and I look at this acid, there's the acid. This carbon, there is a carbon. And this methylene is that methylene. All right. Do you see what needs to attach to each other? Okay. I have right here this methylene, which is right there. And I have this carbon, which I said was this one. So that has to attach to this. Okay. What is missing when I get over here? What leaves? A phosphate. So that phosphate is nowhere to be found. Okay. If this carbon is that carbon, okay, then it has to be double bonded to an oxygen, right? Is this carbon double bonded to an oxygen? No, but is it attached to an oxygen? Okay. And so what we have to do is have this oxygen stay here, but this carbon attack there. I have to make this negatively charged. Okay. Phosphates are replaced onto things because they can be good leaving groups. All right. Here I'm going to start with the water. I'm going to take off the proton. I'm going to have a hydroxide attack the phosphate. I'm going to kick up. I'm going to kick down. And watch what happens here. This pair of electrons forms the double bond. That's the double bond right here. Got that? But when I make that double bond, this carbon now has 5 bonds to carbon. That's not good, right? So one of the electron pairs has to do something. This electron pair could kick up onto this carbon, make a carbanion, which is a nucleophile. Okay? And then it can attack. What I'm going to show is a single movement. When I attack that carbonyl, what normally happens with attacks at carbonyls? I do what? You have to kick up, right? And then you can do what? You could kick down. But if you kick down, what do you have to have in the product? A ketone, right? So if I kick up and not have a ketone, what do I actually end up with right here? A hydroxide. So I'm going to kick up and pick up a proton. In 1, 2, 3, 4, 5, 6, 7 EVZ steps. All right, I have condensed those two molecules together. All right, and oh my goodness, 3-deoxyarabinoheptanosinate 7-phosphate. If you can say that 1 time slow, I'm proud of you. Okay, much less 10 times fast. All right, again, we're trying to make aromatics. Okay, so we're going to get to a ring structure. All right, that was complex. I didn't want you to be scared about that. This is probably a little bit more than I would put on the exam. Okay. All right. Now, Alex, choose the next volunteer. Sarah, come on down. All right. Here is a reaction. Come on, Sarah. That I would expect that you might be able to do. Okay. From this reactant to going to this intermediate. Okay. Now, what might you do? What are you supposed to do when you get the opportunity to look at a reaction? Look at the beginning and look at the end and see what changed. Okay. Between this one and that one?
Wait, this is intermediate.
Yeah, that's it. You're going to go to the intermediate.
So this leaves.
The phosphate leaves. What else has to leave though?
What?
The phosphate has to go away, yes. Do you see any other difference around there?
Yeah, that is a double.
That's a double bond. Do you see any other difference?
The proton.
Ah. So, what do you think we are going to do first off? If there's a proton that is gone, that has to go away.
Deprotonate.
Deprotonate. Because it's?
All about the
That's right, all about the base.
Okay. Oh, that's lovely.
Isn't it lovely? Now, where do the electrons that was between the hydrogen and the carbon go?
Right here.
Yes. And then what has to leave? Ah, very good. You are done. Go choose someone else. All right. Now, before, I never wrote this intermediate for people, and there's all sorts of things that could have happened, but now writing the intermediate helps you identify what was missing. What was missing was this hydrogen and that phosphate, and a new double bond was formed. All right, now going from this part to this part, it seems we have to get a bond formed, right? And so that's interesting, and then something else changes. All right, and when I look at it, oh, that changes up here. That's no change, no change, no change, no change. A change. Okay. So, come on. Now that you see where places are changed, what might happen?
I've never written on this.
You've never written on
No, I
this? Well,
haven't.
first pick a color that you'd
Let's
like.
keep it going.
Okay.
Okay, so I'm gonna draw a base right here.
Blue, yes. Base, yes.
And then can I just draw it straight to this bond?
Yes, into the double bond, yep.
Okay, and then this double bond has to go up here.
Uh-huh. And then,
This has an OH on it, so there's another double bond that will kick up.
uh-huh. Well, before it kicks up, something has to attack it. What's going to attack it? Ah, so at this point in time, you've lost it. At this, the arrow before you started, you have a negative charge on that
I
carbon.
meant to draw. Right.
Okay, then hit back arrow twice. Oh, and you may have done a bunch of times. Oh, no.
Is it permanent?
Huh. Come on. It's
broke it.
it's all gone. Okay. All right. You said that should happen.
Yeah.
Okay, and then it kicks up.
Yeah, and then it stays there, and
But what happens? It becomes a ketone into a alcohol. So what?
so
It doesn't like to be negative, but
right,
then
so
it can.
be a proton.
Yes.
But I don't know where to draw it next. But
Wherever
so
you
would
want.
Would it be better to draw it as negative first and then picking up? Or does
Either
matter?
way, it doesn't matter.
Okay.
But you should understand it does become negative on the oxygen for just a little while, and then it will pick up the proton. But a negative oxygen, even though it's high in energy, is good. Select someone else.
Becca can go next.
Oh, okay. So we're at dihydroquinate, 3 dihydroquinate. At least now we have a ring structure for the aromatics. Okay, so you get to look at 3 dihydroquinate to dihydroshikimate. I didn't make up the names. Uh-huh. Yes. Perfect. Go ahead and select somebody. All right. That is a elimination, correct? I create a double bond. What kind of elimination did she draw in terms of the mechanism? E2. Come on up. Okay, the 3 dihydroshikimate into shikimate. Ooh. All right, so what is occurring now?
We'll lose a ketone.
Uh-huh. Okay, so look at all your reactions. All the reactants. What reactant do you have that is
NADPH.
an NADPH? And so mechanistically, the NADPH gives what?
A proton.
Not just a proton, a hydride.
That's right.
That's right. And negative things love to be nucleophiles to attack at positive places.
So my original thought was this,
Mm-hmm.
but that could possibly be wrong.
That could possibly be wrong because you're attacking electrons to electrons.
Well, all right.
So the hydride is going to be donated, and the hydride likes to attack something that's partially positive. Do you see anything that's partially positive over here?
That, maybe. No, what you think?
It's not formally because you don't see it written as a positive charge, but there is a bond that slightly the electrons tend to be at one side and not the other.
Phone a friend.
Phone a friend? At what kind of bonding has it that's a little bit uneven? A carbonyl.
So,
Mm-hmm.
what you think?
So the carbon is slightly positive because the electronegativity of the oxygen are pulling all those electrons away. So the hydride negative likes to try to get to the positive carbon.
Okay, so I draw here to here. Or
Mm-hmm.
Yeah.
It goes and it attacks at, oh,
You go, well,
at
that's
this
right,
one.
Yeah, okay.
Because look, here you've got an extra hydrogen and then you have an OH, whereas there you have no hydrogen. I've got to put a hydrogen there.
This
Mm-hmm.
carbonyl. This carbonyl?
Yeah, that carbon.
Okay, yeah.
Yes.
All right.
Okay, and then the electrons kick up,
Mm-hmm.
and then
This
it
is a
will
proton, though.
pick up a proton.
Yeah, well, correct.
Yeah.
We'll just go here.
Good. Perfect.
Okay, beautiful.
Beautiful.
Thank you.
Absolutely beautiful.
Don't like you.
And now you get to select someone else. Unfortunately, you can't select her because she's gone today. And we now have Chap17. All right. Okay, so wait. Yeah, she's done it. Who has not had the opportunity yet? Put your head down really, really hard. Really. Here's one. How about over here? Anybody else? There's one. One in the back. Okay, come on up. All right, looking at this reaction, could anybody tell me the class of enzyme that is going to do this reaction? What is the class of enzyme? Okay, we're looking at the end from the beginning. Oh, sorry. And we see like the ring is totally the same, right? We just have a change right here. Around that oxygen, something's missing, right? And then we have something added to it. What kind of enzyme would you classify this reaction as? A kinase. I think you have to, yeah, I think you need to. Yeah, take that proton off, good. And then you'll have that negative pair of electrons that's on the oxygen. You can attack. Mm-hmm. Yes. And then that bond that's between the phosphorus and oxygen, it will then put the electrons onto the oxygen. That's how we show that bond breakage.
How do I show it?
So the electron between the phosphorus and the oxygen, the electron just goes onto the oxygen.
Okay.
Perfect. Perfect. All right.
Who's next?
Who's next? That's your choice. Shikimate 3-phosphate. Okay. All right. Raise your hand if you haven't gone yet. Got 3 possibilities. 4 possibilities. Okay. All right. You can sit down right this very second because this is a very odd reaction. Okay. This hydroxide and this PEP. What is PEP? Phosphoenol pyruvate. What pathway? Glycolysis, or you could also say gluconeogenesis. This oxygen is going to get attached to this carbon. Okay, a phosphate is going to leave. This reaction actually goes through several intermediates. It's complex, so this is not something that I would ask you to do. Okay, I would ask you to be able to identify that this was from glycolysis or gluconeogenesis. All right, in this case, a reaction that I'm going to teach you is going to happen that we need to know about. Okay, so if I take a look at this, answer me this, Batman. Did anything happen or change around this carbon? No. How about this one? No. How about this one? Yes, there's something changed. Okay, what is not written but understood to be there? How many? Okay. around this carbon, is there anything that is not written that should be there? No. There is kind of a change right here, right? Okay. What about here? Is there a change in terms of the number of things that are bound to it? No. Is there a change here? Definitely. What's going on right here that's different? The whole phosphate's gone. Okay. So, on this side, if I take a look, that phosphate's gone, right? One hydrogen. No hydrogens. How many hydrogens? One. Okay. So, what do you think we're going to do? Use a base. Okay. And so I find a place where there's more hydrogens than there should be. Let's deprotonate it. Okay. I'm going to take this pair of electrons, and the only place that it could go is right here to form the double bond, right? But that means that this carbon has issues, right? Too many. So what is it going to do? It's going to do this, and that falls off. At the end, I created more double bonds than there were at the beginning, correct? This kind of reaction is called an elimination. Okay? It is also an E2, but an E2 we know of as this. Leaving group, proton. Proton gives that falls off, the leaving group goes, right? Well, that didn't happen here, okay? Here, I had let's see, 1, 2, 3, 4. Leaving group, I had the proton here, okay? And what happened is, well, what had happened was, that leaves. This is called an E2' reaction. The leaving group is connected through a double bond. It's not directly next to the deprotonation, but it's connected through a double bond. So this is an E2' reaction. So you could describe for me an E1 mechanism, an E2 mechanism, an E2' mechanism, an SN1 mechanism, and an SN2 mechanism. If I drew all the arrows and I showed you a reaction, you should be able to classify whether it is one of these 5 different kinds of reactions that we see. Okay? Where would you might do that? Oh, okay. Joy, joy. Happy, happy. But a mechanism question where you don't have to write any mechanistic arrows. All right. At this point in time, all right, we're going to have cores mate. Cores mate is a branch point, okay, where we can make phenylalanine and tyrosine from that. But what happens here is a really kind of also complex reaction. All right, well, what in the world happens here? Check this out. This carbon has to be attached here. So what if we do this? And I push that there and make that connection because I see that in the product. And do you see what happens to this double bond? It moves. So I'm going to move it. Okay. And then I'm not connected to an oxygen anymore, but I have to have a double bond. That kind of creates a little circle there, doesn't it? Which happened first? Okay. Not enough information. Because if one happens, the other one has to happen, then the other one has to happen, and chicken and the egg, right? And so I would not expect you to understand a mechanism at this moment in time, but you can see where things are differentiated. All right. Branch point, we can go two different routes with this next molecule. So I'm going to show the two different routes. Okay. Both of them lose a CO2, so that's called a decarboxylation, right? One of them has an electron acceptor, so it's going to be an oxidative reaction, so it's an oxidative decarboxylation. All right, the other one is going to have a decarboxylation and a dehydration because I lose water. Okay, in this case, that hydroxide will pick up a proton in terms of the water going. So what in the world happens? If by chance I have an electron acceptor, okay, I can do this. Create the CO2 that falls off. I can then create the double bond inside the ring, and here I can create the movement of the electron, and then I have to have a leaving group. Since I have an electron acceptor, the hydride can be the leaving group and be accepted in the NAD+. Alternatively, I can decarboxylate. I can allow the electrons to go form the ring. I can form the double bond movement, and in this case, I can have that as the leaving group. All right. The driving power of this is the formation of this ring. What is the special designation of that ring? It is what? It's what? It's what? It's a aromatic ring, and that conjugation is very, very stable. All right, here, what amino acid might I make from this one? Tyrosine, and then this one would be phenylalanine. How close are we to an amino acid? Pretty close. Because what kind of molecule has this arrangement? What do we call it? An alpha keto acid, right? And if I react an alpha keto acid with what kind of enzyme? Transaminase, I substitute any double bonded carbonyl into an amine. And I've created my two amino acids. Okay. Course mate not only is a branch point for the two tyrosine and phenylalanine, but it also is the branch point to create tryptophan. All right. In this particular reaction scheme, all right, glutamine will give off to make glutamate, so an amine comes off. I get an amine attached. Then you have no idea what PRPP is yet. We'll introduce that later, so I'm not going to be, that's not going to be important for you to understand. The isomerase, not a bit that you need to understand. You should be able to pronounce that correctly. No. As we go down through the entire synthesis, there is not much that is not extremely complicated in this particular synthetic pathway. Multiple step, but what I want you to understand is that once we get to course mate, we can go to phenylalanine, tyrosine, and to tryptophan there. I won't ask you about the synthetic pathway of this. All right. There are a couple of strange and odd things I wanted to show you in terms of structures. One is a methyl carrier, and we're going to show you two methyl carriers as part of this process. You know, a kinase is something that will bring a phosphate and transfer it around. That happens a lot. But there are a lot of other places in which we need to add a methyl group. Well, methyl groups aren't really great leaving groups, or they're not things that we think about being transferred very easily, but we're going to show you two major methyl carriers because the structures allow that to actually happen. One is adenosylmethionine and the other is tetrahydrofolate, and we will see them used. Okay. The adenosylmethionine is used primarily to transfer a methyl group itself. The tetrahydrofolate can actually transfer a 1-carbon unit in several different oxidation states. And so if you see adenosylmethionine, you already see methionine, and so you probably should be able to recognize or even draw what methionine is, because that's one of our 20 basic amino acids. And it's going to react with ATP. Okay, when we react with ATP, we normally react it at either the alpha, the beta, or the gamma phosphate. Where would you think that the sulfur attacks the ATP? Is it at the alpha, is it at the beta, or is it at the gamma phosphate? Well, if the sulfur actually attacked at any of the phosphates, a phosphate would be attached to it. Is a phosphate attached to it? The sulfur of the methionine actually reacts with the carbon that attaches to all 3 of the phosphates. And so the leaving group is all 3 phosphates. And so it actually attacks at the carbon, and that would be an SN2 attack. But check this out. Nice and happy. It's got pairs of electrons, right? Just like water. Is this molecule happy? No. What is not happy about this molecule? It's got a positive charge sitting on the sulfur. Sulfur doesn't like it, but sulfur reacts kind of like oxygen, right? Can oxygen have a positive charge under certain circumstances? Yeah, it's unstable, but it's there and we see it, so it can exist, although it is very unstable. How in the world do we power this? We break all 3 phosphate bonds. Okay, and so that drives a lot of the power. I get the adenosylmethionine, or SAM. This is so reactive that if anything, including a carbon-hydrogen bond, if anything attacks here, the leaving group creates a non-strained molecule, a neutral molecule. You get adenosyl homocysteine. And so that carbon here becomes able to be attacked with this part of the molecule, a great leaving group. The yellow circle is the activating group to make that carbon reactive. Okay? And so it can transfer a methyl. Now, here the adenosyl homocysteine is a dead end product. So I have to regenerate things that I can use. And in this case, I'm going to add water and get adenosine. I will show you that reaction. Take the proton, attack here. This picks up a proton. That attack of that carbon is a nice SN2 attack. And I get homocysteine. What kind of molecule, if you had to classify this molecule, what category would you classify this as? It is an amino acid. Is it one of your 20? No. It is found in the body, but it is not incorporated into a transfer RNA. There is no codon for this. It is a classic amino acid. It's just an intermediate of a different type of reaction. Now, homocysteine is not used, but what did we start off with? Methionine. And what's the difference between homocysteine and methionine? Methionine, what's attached to this? Not a hydrogen, but a methyl. So we are going to use a second methyl carrier to regenerate. The second methyl carrier, or this one's more a 1-carbon carrier because it can carry methyls, it can also carry aldehydes, is tetrahydrofolate. Tetrahydrofolate is a pretty big molecule, and if you actually take a look at this linker, it can be really large. Okay, but the working part of this are those two nitrogens. A carbon can be attached between those two nitrogens or singly on any one of those nitrogens and be activated such that the tetrahydrofolate is a leaving group. And so in this case, we are going to have the methyl attached to one of those nitrogens, the fifth nitrogen. Okay, and we are going to react it this way. We have the hydrogen, a base, deprotonate, and then the sulfur will attack, and then this will pick up a proton. And we regenerate methionine. And then tetrahydrofolate can then be recharged to do this. All right, this kind of reaction in the adenylyl methionine is going to be important when we do some reactions in the future. All right. In this case, systematically, or to give it a little bit of a picture, methionine reacts with ATP to make the adenylyl methionine. That is what transfers methyl groups in lots of different reactions. I get a dead-end homocysteine that has to be degraded, the adenylyl homocysteine into homocysteine, and then the methyl coming from tetrahydrofolate regenerates the methionine. Another molecule that is out there that is in high concentration within the cell is the glutathione. Look at this molecule and tell me what kind of molecule you would classify this as. Amino acid. Well, amino acids are there, yes, but amino acids connected together is a protein, right? Yes. So when we write proteins, what is the front end of a protein and what is the back end of a protein? The front end is always the what side? The which terminus? Amino terminus. And the back end is always the carboxy terminus, the C terminus. Wait a minute. There's a C terminus and there's a C terminus. But wait a minute. Here's an N terminus. What is wrong with this peptide? Okay, an amino acid and an amino acid connected by a peptide bond, an amino acid and an amino acid connected by a peptide bond. This is glycine, this is cysteine, and this is glutamate. But it's not just glutamate, it's what? Gamma glutamate. What is different about the bond? Which bond is different than what a traditional protein or peptide? Think about glutamate and its structure. Okay. There it is. The bond is connected through glutamate's R group acid and not the acid on the alpha carbon, which most proteins or peptides do. So there's a carboxy group on the alpha carbon that usually is involved in peptide bonds, and there's a carboxy group in the R chain. This puts it together with the R chain. This is not created using transfer RNA and codes that way. What happens is we have two separate proteins that will put this together in a synthetic pathway. Okay. This glutathione is abbreviated GSH. The important part of this is that group there. Okay. Now, why is it important? If by chance I have something that has a sulfur, R, and I have something else that has that sulfur, R, they can react and they can create, and we call that type of bond a what? A disulfide bond. The consequence of a disulfide bond that you may not understand is this. I would deprotonate one, I would attack, and the leaving group is a hydride. Okay? Hydrides are reactive species, okay, and we usually have to have a carrier for them, but what the glutathione is going to be is a reservoir of hydrides, just like NADPH is a reservoir of hydrides that can be used. And so two glutathiones can react, go through a series of reactions and end up like this. And that's designated GSSG. Okay? As a consequence, you form that, plus you get a usable hydride. Now, what's kind of unusual about this is the concentration of glutathione in the cell is about 5 millimolar. All right? And in terms of concentrations inside the cell, that's a really high concentration. Okay? Most things are much lower in concentration in the cell. What this molecule does, it is a source of hydrides to react with peroxides that are there. Now, we don't talk about peroxides too often, but we talk about oxidative damage or oxidative species that are there. It is going to protect against oxidative damage. It doesn't react with the peroxides directly, though. The glutathione will actually react with an enzyme called glutathione peroxidase. In that, there is a selenium ion in the active site, and the selenium ion needs some electrons. When it has an extra pair of electrons, it can react with the peroxide, create a hydroxide, which is non-reactive, and I create an acid form of that. The glutathione will react and displace the hydroxide, and then a glutathione will react with itself, leaving the electrons with the selenium to react with oxidative species. And so the glutathione works through this peroxidase to allow us to react these reactive species. All right. Another species that we are going to see in terms of synthesis that have nitrogen in them is heme. Okay? And heme, we've seen all sorts of hemes. We know that they're in cytochromes, in hemoglobin, in myoglobin, we also have seen the heme in photosynthesis, where we had a heme, but there was no metal center. So they're an important molecule. In this, the heme reaction starts off with glycine plus succinyl-CoA. Tell me, what pathway are we in succinyl-CoA? Citric acid cycle. And we'll create an intermediate, which then these intermediates will be transported out of the mitochondrial matrix, and two of these will create one of these structures that have this ring structure with the nitrogen. We put four of those together to create the ring structure, and then it gets transported in, and then we insert the metal center to get to the heme that we know that is in hemoglobin and myoglobin. Okay? The reaction here of these two particular entities. All right? I do want to show you something because eventually you're going to have a quiz on this. Okay? And it's important, the order of reaction. All right, if I take a look at this in the product, it seems that here's a nitrogen, there's the nitrogen. Here's a carbon, there's a carbon. There's a carbon that is bound across, right? This carbon was the ketone. Well, I like to attack ketones, right? All right, so I'm going to attack this ketone. But where do I attack it? Well, here's a nitrogen, there's a nitrogen. There's a methylene, there's a methylene. Here's a carbon, there's a carbon, and here's the carbon that attacked it. What do we know about this carbon here? It is slightly what? Acidic, because it's alpha-tool carbonyl. What about this one? It's also acidic. Okay. What about this one? It's acidic. And what about this one? It's acidic. Okay. The other connection that I have to make is this nitrogen, which is this nitrogen, to this carbon. Well, that carbon is nitrogen, carbon, carbon, nitrogen, carbon, carbon. Oh, that has to attack. Okay. So I have to make an attack from there to there, and I have to make an attack from here to there. Okay. There is an order by which this can happen. I can do red first, or I can do blue first. Which one might you think happens first, red or blue? Okay, if red occurs, okay, then what you're going to do is once you attack this, once you attack here, that becomes a hydroxide in the intermediate. If red did first, then if this is a hydroxide, is this acidic anymore? No. Why would there be any reason for this to be deprotonated? Once you've destroyed the ketone, this doesn't become acidic. So you have to attack this one first to make this as a hydroxide in the intermediate, because a ketone having this next to it is acidic, and that's fine. But this amine is still acidic because it can be deprotonated and then attack. So it is blue before red. Okay? That's as much of the clue as I'll give you now. I'll let you work on this particular reaction later. All right. Heme is important. Why? We use it a lot. That's good. It carries what? Carries oxygen. That's important to us. So heme is located in what cell? Red blood cells. Okay, that's good. And so when people do blood tests, one thing that they look at is a molecule called bilirubin. Okay. Well, bilirubin is a process of heme being what? Broken down. All right. If heme's being broken down, that means your red blood cells are being broken, and that may or may not be a good thing, okay, in which your red blood cells are being turned over very high, and a higher level of bilirubin in your bloodstream might indicate that there's something bad going on, okay? So this is something that is in one of the normal blood tests. When you are an infant, okay, you can also have this as an adult, but if you have a lot of red blood cells that are being broken down very, very quickly, what color does it say bilirubin is? Yellow. And if you turn yellow, what disease that you have? Jaundiced. Okay. And what's the treatment? Light. Why in the world do you think light will help break down that bilirubin molecule? Why would light even interact with that molecule? Not vitamin D. Okay, look at the organic molecule. What does an organic molecule have to really kind of interact with? Conjugation. And it absorbs that energy, making it more reactive. It makes it more reactive, it will break down faster. And that's why the light is given as a treatment option. Okay, and if you take it down the pathway, you end up in two different places. One, urine. What color was urea? Clear. But people thought it was yellow, right? And what color is this other stuff? And where do you get it? That molecule. Okay, there's where the color comes from. All right. And so what we do is we indicate or we look for the presence of the breakdown products to see if your blood is healthy. All right. The quiz. How many questions do you have to answer? 7. But there are some that you must answer, right? Okay. Please answer the questions in order of whatever. So if you answer 1, 3, 5, 7, put them 1, 3, 5, 7. Don't put 7 and then 3 and then 1 and then 5. Someone did that. That's just it makes it harder to grade. So.